So this is the third and final lecture for the special senses chapter. This is going to be covering hearing and balance. So hearing and equilibrium are two very closely connected senses. So the transduction of sound vibrations by the ear's sensory receptors into little electrical signals is going to be about 1,000 times faster than the response to light by the eye's photoreceptors. So this is a very rapid response that we're eliciting with these particular receptors. The ear is also going to be where we have the receptors for equilibrium. This is also known as balance. And we can divide the ear into three individual regions. We've got the external ear, which you can see kind of here in yellow, the middle ear in blue, and then the internal ear is going to be in this, I guess, pink color that we see there. So, looking at the external ear for starters, we can, again, then to break this up into three parts. We've got the auricle, which is the flappy bit of ear that you actually see on the side of your head. This is made up of elastic cartilage, and it's going to be shaped kind of like the flared end of a trumpet. And this is going to be mainly used to capture sound. It's also, handy for decorating with various pieces of jewelry. So the rim of the helix, of the auricle, is called the helix. This could be up here. And then the inferior portion of the ear, of the auricle, is called the lobe. And this is the thing that a lot of people get pierced. We then have the external auditory canal. And this is basically just a curved little tube that's going to be about 2.5 centimeters long. And this is going to lie within that temporal bone. And this is what is responsible for transmitting sound into the eardrum. And then last but not least, we have the tympanic membrane. This is also called the eardrum. And this is going to be this real thin, kind of semi-transparent membrane. It's going to serve as a partition between the external auditory canal and the middle ear. So this tympanic membrane, this is going to be stretched really tight across that ear canal there. And this is going to essentially vibrate like a drum. And his vibrations are then going to be then transmitted and move and cause movement in the little tiny bones that we see in the inner ear. And this in turn is going to send vibrations through to the inner ear, where we will then convert them into electrical signals. Additionally, in the external ear, we have these things called ceruminous glands, and these are going to secrete cerumen. And this stuff is really important for protecting that ear canal and that eardrum. So you may have guessed that cerumen is actually just earwax, which is gross, but important. Moving further into the ear, then we then come to the middle ear. This is going to be a small little air-filled cavity that's found within the temporal bone, and this is going to be lined with epithelium. This is going to be separated from the external ear by that tympanic membrane, and from the internal ear by a thin bony partition that's going to contain two small openings. And these openings are called the oval window and the round window, and we'll talk more about how these are used in auditory transduction in a little bit. So extending across the middle ear, which we can see here, and then attached to either end by various ligaments, are going to be three small bones. These are actually the smallest bones in the body. These are called auditory ossicles, and these are going to be connected by synovial joints. So the middle ear is going to contain those three auditory ossicles. We've got the malleus, the incus, and the stapes. These are so named because of the way they look. So malleus is, I think, Latin, I believe. It looks like a hammer. It's kind of like a mallet. The incus is named after an anvil, and then the stapes does really very much look like a stirrup. So sound vibrations are going to be transmitted from the eardrum through these three little bones, and that is going to cause pressure to occur and to be put on our little oval window. So the handle of the malleus, of that first bone there, that first auditory ossicle, is going to be attached to the internal surface of the tympanic membrane. And then the head of the malleus, what would be like the hammer part, I guess, is going to actually then articulate with the body of the incus. So it really does look kind of like a hammer pounding on an anvil. The incus is the middle bone in this series, and this is going to then articulate with the head of the stapes, and then the base, or like where you would put your foot in like a stirrup. So the foot plate of the stapes, this is going to fit really nicely into that oval window. And so then directly below our oval window, we can see here, this is going to be another little opening, and this is what we call the round window. And this is going to be also enclosed by a membrane that's called the second tympanic membrane. And we'll talk more about this in a minute. The anterior wall of the middle ear is going to contain an opening that basically leads directly into the auditory tube. Right? This is also commonly called the eustachian tube, which is really important. So this auditory tube is going to extend from the middle ear into the nasopharynx. So this is going to be like in the back of the throat, basically, where your nose and your throat kind of connect. That's the nasopharynx. And this auditory tube, this eustachian tube, is going to be really important for regulating air pressure in the middle ear. So normally the eustachian tube is going to be closed at its medial or pharyngeal end. So the end is closer to the throat. However, during things like swallowing or yawning, it's going to open, and this is going to allow for air to enter or leave the middle ear until the pressure in that middle ear equalizes to that what we see in atmospheric pressure. So when the pressures are balanced, this tympanic membrane is going to vibrate freely, and all the sounds are going to happen normally as that sound wave strikes it. However, if we have built up pressure, or if these pressures are not equalized, this can cause really intense pain, as well as hearing impairment, ringing in the ears, and sometimes even vertigo. So this is like if you dive down deep in a pool, that kind of pressure that you feel building up in your ears, that's from a non-equalized pressure in that inner ear there. Same when you take off in an airplane. So the auditory tubes are also going to be a really fantastic route for pathogens to travel from the nose and the throat to the middle ear. And this is going to be the most common cause of ear infections. So this is why little kids so frequently get ear infections, essentially, is because these little auditory tubes are going to be angled slightly differently until they fully grow into their normal size. And so this is going to cause for a buildup of a lot of gross infection and grossness. So that's one of the reasons that little kids typically have often have so much trouble with ear infections. All right, that brings us in to the inner ear. This has a few more parts to it, so this is a little more complicated. So the internal ear is called the labyrinth, and the labyrinth is a fancy word for like a maze, and that is very aptly named because it is quite the maze in there. So anyhow, the internal ear is going to contain the cochlea, and this is going to translate vibrations into neural impulses that the brain is then able to interpret into sounds. The inner ear is also going to contain these semicircular canals, and these are going to work with the cerebellum to help provide things like balance and equilibrium. So the inner ear is also called a labyrinth, and this is because it's a very complicated series of canals. Structurally, it's going to consist of two main divisions. We've got an outer bony labyrinth, and then this is going to enclose an inner membranous labyrinth. And that bony labyrinth is going to be basically just a series of cavities that we have in the petrous portion of the temporal bone. And this can be divided into three main areas. We've got the semicircular canal, two semicircular canals, and the vestibule. Right? And both of these are going to contain receptors for equilibrium. And then we also have the cochlea. This is going to contain receptors for hearing. So the semicircular canals, the vestibule, and the cochlea. Those are the three divisions of that bony labyrinth there. The bony labyrinth is going to be lined with periosteum and is going to contain a fluid called perilymph. All right, this fluid is going to be chemically very similar to what we see in cerebrospinal fluid, and is also going to surround that membranous labyrinth. The membranous labyrinth, on the other hand, is going to be basically just a series of sacs and tubes that are sort of going to lie inside and have the same general shape as what we would see in the bony labyrinth. So the membranous labyrinth is going to be lined with epithelium and is then going to contain a fluid called endolymph. Right? So the vestibule the vestibule is going to constitute the oval sort of central portion of the bony labyrinth that we see here. And then the membranous labyrinth that we see in the vestibule is going to consist of two sacs called the utricle and the sacculae. So then projecting upwards and posteriorly from this vestibule, we're going to have these three bony semicircular canals that you can see here. All right? And each of these is going to be arranged at about a right angle to the other two. So that anterior to the vestibule is going to be where we find the cochlea located. And this is going to consist of a bony spiral canal that makes almost 3 full turns around a central bony core called the modiolus. Now, if we take a cross section through the cochlea, we're going to see that it's divided into 3 main channels by a partition that's kind of shaped like a Y. So we can see our little Y shape there with our little tail. So the channel above our bony partition, this is called the scala tympani, I apologize. This is called the scala vestibuli that we see here. All right, and this is going to end at the oval window. The channel that we find below this Y shape is called the scala tympani, and this is going to end up at the round window. So the scala vestibuli and the scala tympani are both going to contain perilymph and are both going to be completely separated, except for this tiny little opening that we see at the apex of the cochlea right here in the middle. This is called the helicotrema. Right? This is where any kind of this perilymph fluid essentially is going to make a U-turn, essentially, when we're trying to have any sort of transmission of vibrations or sound. Now, the third channel that we have in the inner ear is the cochlear duct. Right? So the vestibular membrane is going to separate the cochlear duct from the scala vestibuli, and the basilar membrane is going to separate the cochlear duct from the scala tympani. Resting on our basilar membrane is going to be the spiral organ, which is also called the organ of Corti. So this is going to be a coiled sheet of epithelial cells, and this is going to include various supporting cells, and importantly, about 16,000 hair cells. These are going to be the all-important receptors that we have for hearing. So at the apical tip of each of these little hair cells is going to be these little stereocilia. Right? And these are going to extend into the endolymph that we have in the cochlear duct. And then projecting over and in contact with, like what we can see here, and in contact with those little hair cells of our spiral organ, we have something called the tectoral membrane. And this is going to be a very delicate and sort of flexible gelatinous membrane that's going to come into contact with those hair cells. Now, that brings us then to the various physiological processes that are involved in hearing. So the first step for the whole process of hearing is going to be that the auricle is going to direct sound waves into that external auditory canal. Mm-hmm. Sound waves are then going to strike that tympanic membrane, and this is going to cause it to vibrate back and forth. And this vibration is going to conduct from the tympanic membrane through those little ossicles. All right. Now, it's going to move through the malleus, through the incus, and then through the stapes. And the stapes is then going to move back and forth as a result of this vibration, pushing on the membranes of the oval window. And it's going to push this in and out. All right. Now, this movement that we have of this oval window is going to set up a fluid pressure wave in the perilymph and of that scala vestibuli. All right, so it's sending out basically pressure waves, setting up a wave of pressure through the scala vestibuli's perilymph. It's like a wave machine, kind of. Then the pressure waves in the scala vestibuli are going to be transmitted are going to be transmitted to the scala tympani and are eventually going to make it their way to the to the round window, and this is going to cause it to sort of bulge outward into the middle ear. All right. So, as the pressure waves that we have set up in the scala vestibule and the scala tympani, these are going to cause a deformation in the walls of the inner ear. All right. These are going to then push on the vestibular membrane. It's going to push it back and forth, and it's going to increase and decrease the pressure of the endolymph that's on the inside of that cochlear duct. Now, the pressure wave fluctuations of this endolymph are then going to cause that basilar membrane to move just ever so slightly, and this in turn is going to move those little hair cells of the spiral organ against that tectoral membrane. So it's the bending of these hair cells that's essentially responsible for the production of these receptor potentials that will eventually lead to the generation of nerve impulses in the cochlear nerve fibers that your brain will then interpret as sound. So, with regards to auditory transduction, these inner hair cells are going to transduce mechanical vibrations into electrical signals. All right. So as that basilar membrane vibrates, those little stereocilia that we have at the apex of those hair cells are going to then bend back and forth and are going to slide against one another. This is going to trigger mechanically gated cation channels that we have in the membrane of that stereocilia to open. And opening is going to allow for cations that we have in the endolymph to enter into the hair cell cytosol. These cations are primarily going to be potassium ions. So as these various cations enter that cytosol, they are going to produce a depolarizing receptor potential. Additionally, something called a tip-link protein is going to connect a mechanically gated cation channel that we find in the stereocilia to the tip of its taller stereocilia neighbors. We'll look at that here in just a second. So now moving forward, we need to talk about the actual act of auditory transduction. So we've mentioned these hair cells, and when these hair cells that we have are at rest, the little stereocilia that we have on the tops of them on their apical surface are going to point straight up. And the cation channels that are associated with them are going to be partially open. This is going to allow for a few measly little potassium ions to enter the cell. And this is going to cause a weak depolarizing receptor potential. Right? So this weak depolarization is going to spread along the plasma membrane, and it's going to open a few of these little voltage-gated calcium channels that we have at the base of the cell. All right. So as a result of this, there's going to be a small amount of calcium that's able to enter the cell, and it's going to trigger exocytosis of just a very few synaptic vesicles that are going to contain neurotransmitters. So this low level of neurotransmitter release is going to generate a low frequency action potential in the first order auditory neurons that are going to synapse with those little hair cells. Now, that's when they're at rest. However, when some sort of vibration occurs, like a sound happens, you hear a sound. When this vibration happens, there's going to be vibration of that basilar membrane, and this is going to cause those stereocilia to bend towards the tallest stereocilium. And those little tip links are going to stretch, and this is going to pull open. This is going to tug on that cation channel. This is going to cause that cation channel to swing wide open. All right, so we're allowing for a larger amount of potassium to enter the cell. This in turn is going to cause a much stronger depolarizing receptor potential. This larger depolarizing receptor potential is in turn going to cause the opening of more of our little voltage-gated calcium channels This is then going to trigger the release of a larger amount of neurotransmitter. And this increase in neurotransmitter release is going to generate a higher frequency of action potentials that we have in our first order auditory neurons. Now, when vibrations of that basilar membrane cause the stereocilia to bend away from the tallest stereocilium, the tip links are going to become slack, and all of those little cation channels are going to then become closed. So because potassium is unable to enter those little hair cells, the cell is going to become more negative on the inside compared to when it's at rest. And this hyperpolarization of the cell is going to result in a hyperpolarizing receptor potential. This hyperpolarization is in turn going to result in the release of almost little to none of that neurotransmitter. And our first order auditory neurons are going to generate very few action potentials. Now, that's how it all works. A functional or a real life application of this whole process is with a cochlear implant, which is just, I think, mind-boggling that we have the technology to do this. So a cochlear implant is just basically a device that is there to translate sounds into electrical signals that your brain can then interpret and do something with, which is, again, just truly mind-blowing. So these kinds of devices, these cochlear implants, are really useful, very helpful for people with deafness that's caused by damage or malformation to the hairs, to little hair cells that we have in the cochlea. So there are a couple of different parts to this cochlear implant. We've got the external parts of the cochlear implant. These are going to be the microphone, which we can see here. This is a little kind of clear, this little clear bit in this one, and then the sound processor, which is going to be this bit right here. All right, and then we also have something called the transmitter, which is back here. So the microphone is going to be worn around the ear, and this is going to pick up the sound waves, and that sound processor is then going to convert that sound wave into electrical signals. Then we have a transmitter, and this is going to receive signals from that sound processor, and is going to pass them to like a little internal antenna that we have inside the brain. We then in the inner ear, and in the brain actually. So the internal parts of this cochlear implant are going to be the internal receiver and then the electrodes. So the internal receiver is going to relay signals that are coming from that transmitter to the various little electrodes. And these electrodes are going to be implanted in the cochlea. From there, they are able to trigger nerve impulses in those little sensory neurons that we are in the cochlear branch of that vestibular nerve, in that vestibulocochlear nerve, So these artificially induced nerve impulses are then able to propagate over their normal pathways to the brain. And this perceived sound is going to be relatively crude in comparison to what somebody with normal hearing would have. However, it is going to allow for some degree of hearing, and it provides a sense of rhythm as well as loudness. It also can provide information about certain noises, However, such as those like made by telephones, ringing, automobiles driving by, etc. You could also get some indication of pitch and cadence of speech. So you are able to hear, but it does things like talking over a phone line are still potentially difficult. So that's how that works. That's how we have a machine essentially that can translate sound into nerve potentials, which is just truly amazing to me. Now, as far as the auditory pathway is concerned, we're not going to get too in-depth here, but the cochlear nerve fibers are going to form the cochlear branch of the vestibulocochlear nerve. And these axons are going to synapse with neurons in the cochlear nuclei that we find in the medulla oblongata. The impulses are then going to travel to the medial geniculate nucleus of the thalamus, and are then going to end up in the primary auditory areas of the cerebral cortex that we find in the temporal lobe. So that is how hearing works. Very cool stuff. That then brings us to the whole process of equilibrium, also known as balance. So coming back to this inner ear and the structures that we associate with the inner ear, and how these relate to equilibrium. For starters, we've got the vestibule, right? We can see over here, this part right here, it's the vestibule. And this is going to be the oval, sort of central portion of that bony labyrinth. The membranous labyrinth in the vestibule is going to consist of two sacs called the utricle and the sacculae, and these are going to be connected to two by a small duct. Right. Then projecting superiorly and posteriorly from the vestibule are going to be the three bony semicircular canals that we can see here. Each of these is going to lie at approximately a 90-degree angle to the other two. So based on their positions, they are going to be named anterior, posterior, and lateral semicircular canals. And both the anterior, posterior, and posterior semicircular canals are both going to be vertically oriented, while the lateral semicircular canal is going to be horizontal in orientation. So at the end of each of these canals, we're going to have this sort of kind of bulbous sort of enlargement here, and this is called an ampulla. Excuse me. And the portion of the membranous labyrinth that lies inside of these bony semicircular canals are called the semicircular ducts. So these structures are all going to be connected with the utricles, with the utricle of the vestibule. So equilibrium is also known as balance, and it's going to come in two main types. We've got our static equilibrium, and this is going to be the maintenance of the body's position relative to the force of gravity. And then we have dynamic equilibrium. All right, and this is going to be the maintenance of the body's position in response to a sudden movement. So the vestibular apparatus is going to be the organ that's responsible for to help maintain the various organs that maintain equilibrium. And these are going to include the saccule, the utricles, both of which are considered to be otolithic organs of the inner ear that we'll talk more about in a minute. And then these semicircular canals are also part of the vestibular apparatus. The otoliths, part of the otolithic organs there, are going to be calcium carbonate crystals. So the walls of the utricles and the sacculae are also going to contain something called a macula, and there are going to be two macula are going to be receptors for the static equilibrium that we have. So looking at this otolithic organ, the two otolithic organs are going to be the utricles and the sacculae. And attached to the inner walls of both of these are going to be a small thickened region called the macula. The two maculae, which is plural for macula, are going to contain the receptors for linear acceleration or deceleration, as well as the position of the head, so head tilt. So this is what's going to be responsible for detecting that change in movement when you're in the car and somebody slams on the brakes, or if you tilt your head from side to side. The macula are going to consist of two types of cells. We've got hair cells, which are going to be the sensory receptors, and then the various supporting cells that are going to be like the backup singers. They're there to support and nourish, etc., physically, emotionally, structurally support our hair cells there. The hair cells that we have on the surface cilia, hair cells are going to have stereocilia on their surface, and these are going to be graduated heights, kind of like the Verizon bars, right? And then there's going to be also one kinocilium, and this is a conventional cilium that's going to extend beyond the longest of the stereocilium. So this is the longest one here, and it's not a stereocilia technically. So just like what we see in the cochlea, those stereocilia are also going to be connected by little tip-link proteins. So connectively, the stereocilia and the kinocilium are going to be known as a hair bundle. So scattered among the little hair cells are also going to be columnar supporting cells that are probably going to secrete the sort of thick gelatinous glycoprotein layer that's called the otolithic membrane. And this is going to rest on those hair cells. So we have a layer of dense calcium carbonate crystals that are called the otoliths that are going to extend over the entire surface of that otolithic membrane. Now, as far as our looking at this, at how this works then. So the macula of the utricles and the sacculae are going to sit perpendicular to one another. All right, so kind of like a T. So when the head is in an upright position, the macula of those utricles is going to be oriented horizontally, and the macula of the saccules is going to be oriented vertically. Now, because of these orientations, the utricles and the sacculae are going to have very different functional roles. The utricles are going to respond to linear acceleration or deceleration. That's going to occur over a horizontal direction. So like when you're in the car and somebody, taps the brakes or slams on the brakes, you're going to feel that slowing down or speeding up. The utricles on the other are also going to respond to when you tilt your head forwards or backwards, like when you nod yes, like what we see this guy doing right here. The sacculae, in turn, are going to respond to linear acceleration or deceleration that's going to occur in a vertical direction, like that weird kind of jolty sensation when you get when you're going up or down in an elevator. That's being detected by the sacculae. So because these otolithic membranes are going to sit on top of the macula, if you tilt your head forward, the otolithic membranes, along with the actual otoliths themselves, are going to be pulled by the force of gravity. It's going to cause it to slide downhill, so to speak, over the hair cells that are going, and it's going to slide in the direction of the head tilt, or the head bending, causing the bending of those little hair bundles. However, if you were to sit upright in a car and then, suddenly hit the brakes, these little otolithic membranes would lag behind or lag ahead of your head movement. And this is due to the whole process of inertia. This is going to then pull on those little hair bundles and make them bend in the opposite direction. The bending of these little hair bundles in one direction is going to stretch the tip lengths, which is going to cause them to fully open cation channels, producing a depolarizing receptor potential. This is going to, and then alternatively, if you bend them in the opposite direction, this is going to close those cation channels and produce a hyperpolarization. And your brain is going to then be able to interpret this as a slamming on the brakes or a slamming on the gas sensation, essentially, for your movement there, for your equilibrium. The otolithic membrane is going to sit on top of the macula, like we mentioned, and movement of the head is going to cause gravity to move it down over the hair cells. So as these little hair cells are then going to synapse with neurons in the vestibular branch of the vestibulocochlear nerve, which can then be transmitted to the brain. Remember, for the other parts of that inner ear, we also have these semicircular canals. These are going to be responsible for the dynamic equilibrium portion of balance, essentially. The ducts are going to lie at right angles to each other, and this is going to allow for rotational acceleration and deceleration to be detected. So an ampulla that we see an ampulla in each of these canals is going to contain something called a crista, and this is going to be along with a group of hair cells. So covering that crista is going to be a mass of sort of gelatinous material called the cupula, and then movement of the head is going to have an impact on the endolymph as well as those little hair cells. This in turn is going to generate a potential that is going to lead to nerve impulses that are then able to travel again along that vestibular branch of our vestibulocochlear nerve. Now these semicircular canals, when the head rotates, like when you're spinning in circles, the attached semicircular ducts and those little hair cells are going to move. However, the endolymph within the ampulla is not going to actually be attached to anything, so it's going to lag behind the rest of your head due to the whole process or the principle of inertia. So the drag of the endolymph is going to cause the cupula and the associated hair bundles that project into it to bend in the direction opposite to that which the head is moving. So as if the head continues to move at a steady pace, then the endolymph is going to begin to move at the same rate as the rest of the head. And this, in turn, is going to cause the cupula and its embedded little hair bundles to stop bending and to return to their resting position. However, once the head stops moving, that endolymph is temporarily going to keep moving in the direction that you were just spinning due to, again, inertia. This, in turn, is going to cause that cupula and its little hair bundles to bend in the same direction as the preceding head movements. So at some point, that endolymph is eventually going to stop moving, and the cupula and its associated hair bundles are going to return to their resting unbent position, right, at which point you will cease to be dizzy, essentially. It's important to note, too, that the bending of these little hair bundles in one direction is going to depolarize the hair cells, while bending in the opposite direction causes hyperpolarizing of those cells. Now, looking at the neural sort of connection to all of this. Most of vestibular branch fibers that we see in the vestibulocochlear nerve are going to enter the brainstem and are going to terminate in the medulla. Right? However, there are going to be a remaining few that are going to enter into the cerebellum. Right? So various pathways between the vestibular nuclei, the cerebellum, and the cerebrum are all going to enable the cerebellum to play a key role in maintaining both static and dynamic equilibriums. So here we have a cute little table summarizing the various structures of the ear, as well as links to associated Wiley Plus if you're interested. And that is all I have for this particular lecture. So as always, if you have any questions, comments, or concerns, please feel free to contact me or ask after class. So I'll see you next time.